I'm Andrés Klein. I'm a Chilean biochemist, and I work in Chile currently. I have my own independent lab, Desarrollo. And since, I believe like 10 or 15 years, I got interested on why patients can present with different disease severities. Okay, even though they share the same genetic mutation, in some cases they develop, let's say, a very aggressive neurological disease, while in other cases they don't. And I believe that if we understand why some people are resistant, we can develop novel therapies for the susceptible ones. So conceptually it's a bit different from the classical way in which we study the pathological cascades, in which we study what is affected first and then second and so on. So we're using this different approach, and luckily we're getting some novel, exciting results, still at the very preclinical level, but with a lot of potential. So I just talk about how we are applying this strategy in this particular case. So I show some published data about Gaucher disease and how modeling these diseases in animal models of different genetic backgrounds, we mimic the phenotypic variability that exists in humans. And the advantage of using model organisms is that their genomes are already sequenced and we have access to tissues. So that allowed us, for example, for the case of Gaucher, to identify candidate genes and druggable pathways to treat the neuropathic patients, which currently there is no approved treatment for them. And we beautifully shown using an FDA approved drug called memantine, and now we are willing to push that into the clinic. On the other part, the second part of my talk, I show unpublished data about how we've been modeling Niemann-Pick C disease in yeast of different genetic backgrounds. And again, we mimic or reproduce different severities, which are equivalent of what happens in humans, to map candidate modifier genes. And when we targeted the genes that were identified in the yeast into human cells, we managed to obtain a clearance of the material that usually accumulates in the disease. So it's so beautiful because we started with a very simple organism. We did some validations in flies as well, and lately in humans, and everything is conserved. I guess that very important taking home messages. The first one is treasure your exceptions, because there is so much knowledge in there. We can learn so much from resistant people. So that's the first one. When we don't have access to the humans that are resistant, we can go and take advantage of the genetic backgrounds of the different models organisms and use that to map genes which may serve as therapeutic targets and also biomarkers of severity.